Hi there. This is part two of evolution of ribosomes and the genetic code. I covered quite a bit of the evolution of ribosomes in part one and also some of the genetic code. Today I want to extend some of the discussion about the genetic code evolution. And some of it's a little bit complex, so if you have questions, please let me know. So in the last video, I talked about the genetic code and how generally it seemed to have evolved, starting from these simpler amino acids down here, valine, alanine, aspartate, glutamate, and glycine, and then first moving up along this left-hand side, and then some portions being added going left to right, and then others going from the acidic, the aspartate and glutamate, up into glutamine and asparagine, and then finally this upper row, especially this upper right-hand corner, those were added last. This shows a little more detail, and this is based on the frequencies of those amino acids in ancient proteins. So alanine has the highest frequency of appearance, so the genetic code may have begun here, expanding to valine, glutamate, glycine, and then going up to isoleucine, threonine, across to arginine, and then adding other amino acids through time. But this still presents sort of a confusing picture to me and to other researchers, and probably to you also. So again, if you look at it this way, you can see some structure. There are nonpolar amino acids in orange, polar ones in purple, basic amino acids in blue, and then the acidic amino acids in green. And then a start codon at methionine and stop codons over here on the upper right-hand side. And so comparing that with how the amino acids appeared over time, according to their frequencies in ancient proteins, it still is difficult to discern a clear evolutionary picture. So a few researchers started to rearrange the genetic code in different ways to try and discern some sort of pattern. And so if you separate the genetic code this way, and you start looking at it as far as the class 1 amino acyl tRNA synthetases, those are the enzymes that add the amino acids to the tRNAs, into the class 1s here in yellow and class 2s in blue, somewhat of a pattern is indicated here also. And if you look at this series of rows down here, the ones starting with G, the codon starting with G, you see that for alanine, which might have been the first amino acid, and valine, and glutamine, or glutamate and aspartate, and glycine, you have roughly equal numbers of of class 1 and class 2 amino acyl tRNA synthetases. So this was a clue as to what might have occurred during the evolution of translation and the genetic code. And you can see here, it would be easy enough to move upwards in this direction with the class 1, and then in this direction over here with the class 2 AARSs. Which is indicative of how some of the other analyses have indicated that evolution occurred in this genetic code. Another way that this was analyzed was to look at whether the amino acyl tRNA synthetases approached from the minor groove side of the tRNA or from the major groove side of the tRNA, so from one side or the other side. And then you even get a stronger pattern of evolution in that all of these in this column are approaching from the minor groove side, and all of them here are approaching from the major groove side. And then there's a mixture over here on the right-hand side of the genetic code table. And there are a few that don't seem to quite fit in. That is lysine here, which varies from what side it approaches between bacteria and some archaea. And then tryptophan up here is a bit of an oddball, but in this upper right hand quadrant, you have the most recent arrivals in the genetic code. So it's not uncommon that some differences in the pattern of evolution may have occurred more recently. From some biochemical information and some sequence information, the starting point seemed to be with a class 2 amino acyl tRNA synthetase, and alanine also looks like the most frequent, so the genetic code probably started here, and then you got expansion of the class 2s and then class 1s that approached the tRNA from the other side, and then expansion in the first base position moving upwards, and then finally adding a third base that then filled in and sort of bisected some of the categories over on the right-hand side primarily. Another thing to keep in mind with regard to evolution of these amino acyl tRNA synthetases, or AARSs, is that early on the AARS attached only to the acceptor arm and didn't contact the anticodon down here. So you see here's class 2 very early, probably 4 billion years ago, 3.5 to 4 billion years ago. A little later on, it did develop an arm that contacted the anticodon, but early on, it wasn't attached to the anticodon. So it was basing the reaction of adding the amino acid according to the structure of the tRNA, but not considering the anticodon. And even today, with modern tRNAs and modern class 2 and class 1 amino acyl tRNA synthetases, there are some of those AARSs that do not contact the anticodon. So they're adding the amino acids based only on the tRNA structure rather than the anticodon, which always seemed amazing to me, that they're not even recognizing the anticodon, but then the anticodon is determining what amino acid will be added based on the codon of the mRNA. And here's a phylogeny of group 1 AARSs. You notice that the tRNA is reversed from the previous slide, which had class 2s approaching from the opposite side. And again, very early on, some of the amino acyl tRNA synthetases did not contact the anticodon at all, and it was only later in evolution that they began to recognize the anticodon as well as the structure of the tRNA itself. So this also brings up an important point, that each of the tRNAs has a particular structure that is recognized by different amino acyl tRNA synthetases. So although they all have this typical L shape in their three-dimensional structure, they all have subtle differences in their structure that are recognized by those amino acyl tRNA synthetases. Here's a diagram comparing the evolution of the two, both class 1 and class 2s, and class 2s evolved first, approaching the RNA from one side, and the AARS class 1s evolved just slightly behind those and started to interact with the opposite side of the tRNA. And then you can see that they get larger and they start to contact the anticodon loop and get more complex as well as the tRNAs are becoming a little more complex by that time also. The fact that these recognize opposite sides of the tRNA and initially would recognize similar amino acids means that you can sort of flip the recognition and the anticodon to represent a slightly different, although similar amino acid using a different class of AARS, and that indicates something about the evolution of the genetic code as well. You can rearrange the genetic code in a slightly different way, or actually a very different way, by looking at the codons and the complementary codons, so looking in one direction versus the other direction, and you come out with something that looks, very organized. So this is one where on one set of amino acids is coded by YUN, where Y is a pyrimidine, either a U or a C, N is anything. And in the opposite direction, it's NAR here, and R is a purine, either an adenine or guanine. So organizing it this way, when people first started seeing this, they said, I wonder what the first mRNA looked like. And so they're wondering whether the initial RNAs were double-stranded, and you could read the genetic code in one direction from one part of that strand, and then when it doubled back and became double-stranded, you could read off the other strand in the other direction. These complementary codons that then would encode each of the different amino acids. So that's one reason why people are thinking that, at least early on, mRNAs may have been double-stranded, and you could read in opposite directions off of those two strands. And if you notice on this strand up here, that third position is an N over here, whereas this one in the first position is an N. That is, those bases are ambiguous. And this is one reason why last time I said you can go from a one nucleotide code, meaning that bottom row on the genetic code table for valine, alanine, aspartate, glutamate, and glycine, going from a one nucleotide code to a three nucleotide code, because now you're reading off both directions. So that middle position was the important one. But the other positions, in some case, the first position of the codon was important, but the third one wasn't so important. And in the opposite direction, that means there would be, the first position would be somewhat important, and it could pair with something that wasn't so strict on the other side. And so then that third position is rather variable because it was not strictly pairing with the first position on the opposite strand. I hope that makes sense. And again, I should point out that there are a few exceptions. Lysine, again, is different in archaea than in bacteria, so there's a little difference there. And tryptophan doesn't follow exactly the rule, but that's a very late arrival to the genetic code. I rearranged the previous slide and figure a little bit because I reversed these up here, so it was a little clearer what was going on, or I hope it's a little clearer. So you have one strand going in this direction, YUN again, and in the opposite direction it would be NAR, but then when you look at the codon it would be this way, so you get a fairly clear picture of what's going on in, for instance, leucine and isoleucine, etc. It's a little more muddy when you get to this other side because each one has a different amino acid for that code. But if you look at the genetic code, it makes perfect sense how these are arranged. But anyway, again, this is an indication that originally then you had double-stranded mRNA molecules when the genetic code was very young, and then you had these class 1 and class 2 aminoacyl tRNA synthetases that would charge the tRNAs on opposite sides, and originally some of those amino acids that they would charge were similar, so then you can start flipping these things around within the genetic code to add each one of the amino acids. So because this is still rather difficult to explain in just a few words, and it was confusing to me too, I tried to make more sense out of it by making a diagram like this. And this is the original book version. That is the version from my book, Integrated Molecular Evolution. So here you start out with glycine, which was sort of the way many people thought, and I thought originally. And then that's a class 2 aminoacyl tRNA synthetase in black, and the class 1s are in white. You could then expand into alanine, threonine, serine, valine. But valine would reverse the genetic code, right? So instead of GGN, now we have a change. This would be NGG in the opposite way. So now you're changing a U to a G and reversing that because you're charging it with a class 1 AARS. Since then, I've done a lot more reanalysis of that. And so in 2009, I put out a new paper refining this, but now starting with alanine, because it seems like alanine is definitely the starting point rather than glycine. But the alanine codon GCN, you have a change from a G to a C in that second nucleotide. Then you get a GGN, and you can produce, you can add glycine. So in each one of these symbols, just to explain, the 1 means that's in the highest frequency in ancient proteins. That's its actual frequency down there. There's alanine with the molecular weight. The N there means that it's non-polar. You'll see other ones with P that are polar, or a positive charge or a negative charge. The codon is GCN. The asterisk means that the aminoacyl tRNA synthetase does not touch the anticodon, and the caret there means that it has proofreading function, means that if it makes a mistake, because it's similar to other AARSs, that there's a proofreading function that'll check to make sure that alanine is attached rather than a different amino acid. The last letter down there, POF, means that these amino acids are connected with the biosynthetic pathway for pyruvate. So alanine can be made from pyruvate, and pyruvate can be made from alanine. So they go in both directions. O and F, that's oxaloacetate and fumarate. So you'll notice many of these are coming off things that are around the TCA cycle or Krebs cycle. And so that was a very ancient pathway, and the amino acids could sort of be exchanged. They could either enter the TCA cycle or all the way up to pyruvate, or they could be produced by that pathway, another biosynthetic pathway to make these amino acids. So you'll notice reading through here that many of these are connected in similar pathways. The amino acids that are on the bottom row of the genetic code, these earliest ones, are kind of enclosed in this little gray polygon here. So they're all right together. So that also is fairly consistent with a beginning of the genetic code on that bottom row of the table for the genetic code. And again, alanine is number 1 in frequency in ancient proteins. Valine is number 2. Before I get to valine, let me just explain a simpler one, and that's going from alanine to glycine. All you need to do is have a mutation in that middle nucleotide from a C to a G, and then you have a glycine codon. And so a different amino acid, glycine rather than alanine, could be encoded by that codon-anticodon connection. So those are both being added to the tRNA. The amino acids are being added to the tRNAs by class 2 aminoacyl tRNA synthetases. To go from a class 2 to a class 1, that is from alanine to valine, then you have to look at reverse complements. So a GCN here would be over here in valine, an NGC codon. So because the N is on the ends of each one of those, then those nucleotides on the outside can be almost anything, so you can pair with almost anything. But the middle nucleotide, see, now is changing, in this case, from a C to an A, which becomes a U in this one, because that's the complementary base to that. So that's how you can switch from a class 2 to a class 1. You also have to switch the codon on the opposite strand, really, of a double-stranded mRNA, which was probably what it was in the beginning. And so going through this entire code, this entire chart, then you can make the switches. So from black to black, that is class 2 to class 2, the transition is pretty, or the transition or transversion is pretty simple to see. But going from a class 2 to a class 1 or a class 1 to a class 2, then you have to reverse each one of those codons into a complementary codon and then make the change from there. So the logic becomes a little bit dicey. But then if you look through here, you can see how the genetic code was built up. Here's the frequency 1, frequency 2, 3 with glutamate, 4 is glycine, number 5 is isoleucine, and so on. A few strange things in here. One is arginine is at a higher frequency than you might expect, but it looks like it has evolved to have 2 different codon categories, and so instead of having just 4 codons encoding for arginine, there are actually 6, so it has a larger number. So that one may have increased in frequency primarily by having more codons encoding for it. Also, it seems to have originated from proline codon and AARS. And proline is one of those ones that's somewhat useful in proteins, but you can't use too much proline because it forms kinks in the protein. So proline is probably at lower frequencies primarily because of its function, and there's some selection against having too much proline in a protein. The amino acids with double circles around them, those are those 10 amino acids that were present very early on Earth, and you can see they're among the top 10 or 11 of the amino acid in ancient proteins. The ones with single lines appeared somewhat later, and the ones with dashed lines are the last ones to appear, and you can see those are more complex, and they're on the outer periphery of this diagram as well. 2 groups of amino acids are surrounded by dashed lines. One group is threonine, serine, and proline, and those have homologous tRNAs. That is, the tRNAs that are used for those are definitely very, very closely related. as are their aminoacyl tRNA synthetases. And then the other group is over here, valine, leucine, isoleucine, and methionine. Those also use homologous tRNAs, very, very closely related tRNAs, and evolved essentially as a group. Now, methionine arrived very late in this process as a start codon, as well as just an amino acid within a chain. But it wasn't originally a start codon, and so in some places in here you can also see different start and stop codons that have originated in various places. For example, codons for valine, leucine, and isoleucine, as well as methionine, act as start codons in some systems. And there's also a rare GAG start codon that is a variant of a glutamate codon. Also, in several places, you can see stop codons. The common ones are UAA, UAG, and UGA over on the right-hand side. But there also are others that act as stop codons down around arginine. There's some rare stop codons down there, and some others that appear kind of sporadically in different places in the chart. And then there are a few codons which encode different amino acids, different than the standard. So when we always talk about the universal genetic code, it's fairly universal, but there are some differences in different systems, different organisms. And this diagram connects with the last diagram also, in that, again, you start here at alanine, spread out into valine, glycine, and then aspartate and glutamate, and then move up into isoleucine, over to arginine, and eventually threonine and serine, and the last little bits of the genetic code are up here in the upper right corner with tyrosine, cysteine, and tryptophan. But again, some of these vary. So originally the start codons were all over on this side with the second position U. Now just methionine is the major start codon, and stop codons were pretty much about half of the genetic code consisted of stop codons. Now these three, UAA, UAG, and UGA, are the primary stop codons, but that's not universal either. And again, to broaden this out a little bit, you start here down on the bottom, move generally up on the left-hand side, over a bit, and then up a little bit on this right-hand side, and then finally from left to right to fill in that last upper right corner of the genetic code. And moving back to this diagram, you can see very early on this system was very simple and consisted of still the small subunit, which was primarily carrying the mRNA to the ribosome with the peptidyl transferase activity. The tRNAs were being charged by the class 1 and class 2 aminoacyl tRNA synthetases. The mRNA was probably double-stranded from the beginning because of the tRNAs are charged, and how you have this very regular pattern of self-complementary codons in the genetic code. It looks like it was probably double-stranded, but at some time point it then became single-stranded, probably through its interactions with the ribosome and the fact that RNAs don't stay double-stranded for more than about 7 to maybe 10 nucleotides at a time. So you could only make small peptides from that kind of a system, which was probably the way it started, with small peptides at first. The ribosomes themselves down here, as well as the tRNAs, went through some interesting evolutionary events. The ribosome seems to have been built up by pre-existing pieces that were sort of put together like Legos. So here you start with a simple pattern, and then where those triangles are, you snap on other pieces and other pieces and other pieces. And so it wasn't built up in small little bits, but it was built up of RNA molecules that were already existing and putting them together. Probably introns had a lot to do with putting that together, and there are many ribosomal RNA genes and tRNA genes that still have a lot of introns. And you can see up here that the tRNA was probably put together in the same way, using introns as sort of enzymatic RNAs that would put these pieces together. mRNAs also have introns, but of different types. So ribosomal genes have group 1, group 2, some group 3 or spliceosomal introns, as well as some archaeal introns. tRNAs have primarily archaeal introns, but some have also group 1 introns. A few have group 2 introns. mRNAs have primarily spliceosomal introns that are related to group 2 introns. And so I wrote a paper about this, and this is sort of the final figure of that paper, whereas you have these pieces being joined together in an RNA-centric world to the point where you have larger and larger and larger molecules, and that would be the tRNAs being built up, and especially the ribosomal RNA being built up in pieces. So sort of, as I said, like Lego blocks or something like that. In a DNA-centric world, what has happened then, some of the first introns that were in the ribosomal locus were probably group 1, and the ribosomal locus is filled with group 1 introns in different types of organisms. These can duplicate and move around within the ribosomal locus, and some of these appear to have changed, to have mutated and become group 2 introns, which sort of move around by a different method. But it seems now that they are related one to another. So group 1 introns move around through the sort of like a transposon using an endonuclease, and group 2s move around primarily by reverse transcription, more like a retrotransposon. Eventually you have some of these that are somewhat defective group 2s, and they become the spliceosomal introns. And then there are other types of introns, these archaeal, that seem to have popped in to the ribosomal locus too. So the ribosomal genes, which are in multiple copies, seem to have served as what I call intron nurseries. And I'll talk more about this when I talk about introns later in this video series and in the semester. tRNA also has an interesting past, its evolution. Here's the clover leaf, but if you look at this part, including the acceptor arm and the T domain, or T pseudo UC domain, and the D domain over here in the anticodon, if you compare the sequences, they look similar. So the T and the D domains have some similarities, may have come from the same original sequence. And then the anticodon and then a few nucleotides on the end of the acceptor arm also have similarities. So it's thought that initially you had just the acceptor arm and a T domain. That was duplicated to become this other part of the tRNA, and that became the modern tRNA in the L form, or a clover leaf if you spread it out in 2 dimensions. So by comparing various tRNAs, then it's been determined that the acceptor arm is the most ancient region, and then the T pseudo U C domain is a little younger than that. And then the D domain and the anticodon arm are more recent, and the variable region is even more recent than those. So if you look at how this may have occurred, then the first thing to appear actually was the CCA, but then after that you had an acceptor arm. Then a piece was added on, which became the T domain, or T pseudo UC domain. And then this duplicated. So these were duplicated. They then came together in this form, which when you spread it out, then after you join these together, you come out with the modern look of a tRNA. And all of this probably occurred, between 3.8 and 4.2 billion years ago. And if we look at a scheme, there are two types of protein synthesis ongoing in cells today, including your cells. There's this one over here, which becomes protein synthesis by ribosomes using these different protein and RNA subunits. And then there's non-ribosomal protein synthesis, NPPS, which is more rudimentary. You can make peptides, but the fidelity isn't great. But that's still going on today. So if you go back to the original, you have something that originally would attach amino acids together. So you would have this primordial biosynthesis of dipeptides, and then you could take the dipeptides and join those together with ligases. These are the ligases to form proteins. So you could produce small proteins and peptides, but the fidelity would not be perfect, so that there would be quite a bit of variation in the peptides that were formed. So some organisms would actually be fairly successful, but there would be a lot that would be unsuccessful also. So not the best system for continuing life forms over long periods of time. And as I said, this system still is present in modern organisms. There are these components that contain a condensation domain, an adenylation domain, and a thiolation domain that holds on to amino acids. And they're composed of proteins primarily now, but they may have originally been RNA molecules. And one by one, they can pop together the amino acids to become fairly large peptides. But again, the fidelity of putting those pieces together is not as high. So there's a lot of variation that can be produced. Some of these produce toxins in some microbes that will kill other organisms, and they're hard to keep up on because there is so much variety in the way that those are put together. However, they probably came out of the same system that eventually formed ribosomes. So this may have been the pathway for producing both the non-ribosomal protein synthesis or peptide synthesis and translation. So you start out with a tRNA, and you have a synthetase that adds the amino acid on, and then you have two of these coming together to add the two together. So now you get dipeptides, two amino acids, and then another molecule, a ligase, can put those together, so you can make a string of amino acids into a peptide. And a peptide, again, is a string of amino acids, 50 amino acids long or shorter. So you can see this, depending on how the ligase puts these together, you can have quite a bit of variability in the peptides that are formed. An improvement on this, as far as the diversity and fidelity, would be to put the synthase and the ligase together, so that as these these amino acids are being brought together in peptide bonds, the dipeptides are also ligated together. So you can make longer and longer peptides and eventually proteins. That is, polypeptides longer than 50 amino acids in length. So that can lead to this type of system, non-ribosomal peptide synthesis, which apparently it has. This is one outcome of that type of system. So in this scheme, you start out over here with the synthase, which is making dipeptides here, putting 2 amino acids together at a time. You add the ligase down here to make proteins, peptides and proteins. And then you have this more refined system, which is what is present now, where you have the synthase, which adds the amino acid to the RNAs, and also the ligase that links dipeptides together into longer polypeptides. But you also have this system over here where it's become even more refined with more RNAs, more proteins to become ribosomal polypeptide processes that we know now as translation. So here's a simplified scheme of that, where initially you have just the acceptor arm, which is attached to an amino acid by the aminoacyl tRNA synthetases. Those can be linked together then by synthase as well as the ligase. But then you start getting elaboration of both the aminoacyl tRNA synthetases as well as elaboration of the tRNA, and finally get this protoribosome that does a much better job at linking amino acids together and holding on to the peptide. A further elaboration on that is then the tRNA duplicates in size, or doubles in size, and the ribosome gains these A, P, and E sites. The A site being a duplication of the P site, and it holds onto the tRNA longer. So linking amino acids together is done with higher fidelity and more efficiently. Because of that, the resulting organisms that first develop ribosomes and elaborate tRNAs and aminoacyl tRNA synthetases and RNAs, those organisms become much more successful than the organisms that just have non-ribosomal protein synthesis. And so those are the organisms we see today that are the more successful ones, that have an elaborate translation system that leads to a diverse set of proteins that are formed with high fidelity so that they can be replicated from one generation to the next, to the next, to the next. And this again illustrates that evolution from a fairly simple beginning of some RNAs that probably did other things before they became the ribosome and the tRNAs, etc. And then started to grow, often by joining already formed RNA pieces together to take on new functions, including the peptidyl transferase, the ratcheting of the small subunit, coding of the mRNA and decoding by the by the tRNA, and setting the genetic code by these aminoacyl tRNA synthetases. So now we have, by 3 billion years ago, where we have bacteria and archaea, a very elaborate translation system for making proteins. So that's the end of part 2, and the end of this 2 parts of the evolution of ribosomes and the genetic code. Again, a very elaborate system that evolved only once, starting probably more than 4 billion years ago, and becoming so successful that we only see it in organisms. We see this ribosomal translation system in all cellular organisms that we know of right now. So if you have questions, let me know. I know it's a bit complex, but a very interesting system that built up over the billions of years of evolution on Earth. Okay, so thanks very much for listening and watching, and I will talk to you later.